Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee.
Welcome to the Stack series. Today we are exploring antimicrobial resistance and the post-antibiotic era.
And let me just say right up front, we have an absolute mountain of material to get through today.
Oh, totally. A literal mountain. For this exploration, we are looking at a massive stack of medical journals, some really cutting-edge biotech clinical trials, and a lot of reports from the World Health Organization.
Right, including their newly updated 2024 Bacterial Priority Pathogens list.
Exactly. So our mission today is simple, but, it's incredibly urgent. We need to understand the terrifying rise of superbugs, exactly how these microscopic organisms are outsmarting our absolute best drugs, and the mind-blowing new science that researchers are deploying to save modern medicine.
Things like CRISPR and engineered viruses.
Yeah, the sci-fi stuff. Because this is a discussion about the invisible war happening right inside you and all around you. If you are listening to this right now, this affects you directly. It really is a war. And it's a conflict we've been actively waging for almost a century. To truly grasp the stakes of this discussion, we really have to start by looking back at what we consider the golden era of antibiotics.
Right, the discovery of penicillin.
Yes. Most of us think of Alexander Fleming in 1928 or the introduction of sulfonamides in the 1930s as these absolute turning points in human history.
And they were, right. Think about it. Before that moment, a scratch from a rosebush or a minor paper cut could legitimately lead to a fatal infection.
They absolutely could. Suddenly, these virtual death sentences became minor inconveniences. Humanity felt invincible. But what's fascinating here is that the victory was always an illusion.
Okay, let's unpack this. An illusion. Because from where I'm sitting, antibiotics shaped the entire modern world.
They did, but
You don't have modern surgery, you don't have chemotherapy, you don't have safe childbirth without them. How was it an illusion?
well, because even as we were celebrating the eradication of these fatal bacterial threats, the resistance was already building in the shadows. We introduced penicillin broadly in the 1940s.
Right.
By 1947, we were already seeing the very first documented cases of penicillin resistance in hospitals.
Wow. Just a few years.
Exactly. We thought we had conquered nature, but we had simply triggered a massive evolutionary arms race. And the most pervasive myth we need to dispel right at the start of this discussion is the idea that human beings somehow created antibiotic resistance by inventing these drugs.
Wait, really?
We absolutely did not.
Because the narrative you always hear is that because we overprescribed these pills, we somehow engineered these superbugs in a lab or something. If we didn't invent the resistance, where did it come from?
Resistance is an ancient, completely natural process. We have to remember that antibiotics are, for the most part, naturally occurring compounds.
Oh, like from mold.
Right. Fungi and bacteria have been producing them in the soil for billions of years to compete with each other for resources. It's a microscopic turf war.
So they've always been fighting.
Always. And to prove just how ancient this is, researchers conducted an expedition where they drilled deep into the permafrost and discovered 30,000-year-old bacterial DNA frozen in the ice.
30,000 years?
Yes. This is DNA from long before human civilization even existed. And when they analyzed it, that ancient DNA already possessed the specific genes for resistance to beta-lactams and tetracyclines.
Those are modern drugs, right?
Two of our most important modern antibiotic classes, yes.
So mammoths are walking around, and the bacteria in the dirt under their feet already know how to defeat the drugs we wouldn't invent until the 20th century. That is wild.
It's incredible.
But if they already had the resistance, what exactly did we do to make it a global crisis?
Well, what human beings did was take these natural weapons, mass-produce them in factories, and flood our environment and our own bodies with them. In doing so, we applied an extreme level of natural selection. If you have 1 billion bacteria, and just one of them carries that ancient resistance gene, and you flood the area with antibiotics, you kill off the 999 million susceptible bacteria.
You clear the entire battlefield.
Precisely. And that leaves the one naturally resistant survivor with unlimited resources to multiply and completely take over. We didn't invent the superbug. We just aggressively bred them through sheer evolutionary pressure.
So we basically put them through a global evolutionary boot camp. We forced them to adapt or die, and they adapted.
They absolutely did.
And the ways they actually fight off our drugs are incredible. I was reading through the sources for today's Stack series, and they detail 4 main mechanisms of bacterial resistance. These are essentially master classes in microscopic warfare.
They really are.
So if we are flooding the body with these drugs, How are the bacteria physically surviving the attack? What are they actually doing?
They have developed some incredibly sophisticated defense systems. Let's look at the first mechanism, which the literature refers to as decreased uptake.
Okay, decreased uptake.
Gram-negative bacteria, in particular, are incredibly adept at this. They possess a complex outer membrane that acts as a primary defense barrier, kind of like a fortress wall.
Right.
Now, to let essential nutrients in, they have these microscopic channels built into the wall called porins.
So porins are basically just tiny pores or doorways in the cell wall.
Correct. And antibiotics, especially water-soluble or hydrophilic ones, are designed to use these exact same porin channels to sneak inside the bacterial cell.
Like sneaking in the back door.
Exactly. But when these bacteria are exposed to a hostile environment filled with antibiotics, they sense the threat. They can genetically alter the shape of these porin channels, or they can completely downregulate them.
Downregulate meaning?
Meaning they simply stop producing those specific doorways altogether. A classic example documented in the clinical literature involves a particularly nasty pathogen called Pseudomonas aeruginosa.
Oh, Pseudomonas. That's a bug that causes massive problems in hospitals, right? Especially for patients on ventilators or with compromised immune systems.
Exactly. It's a major cause of hospital-acquired infections. When Pseudomonas aeruginosa is faced with carbapenem antibiotics, which are often considered our heavy-duty last resort drugs,
Right, the big guns.
it simply stops producing a specific porin called OPRD. The antibiotic approaches the cell, looking for the doorway it usually uses to get inside, but the doorway is gone.
Wow.
The antibiotic is locked out, bouncing off the cell wall, entirely unable to reach its target inside.
I love that analogy. It's like the bacteria simply changing the locks on the fortress doors or just boarding up the windows and waiting out the storm.
That's a perfect way to visualize it.
But what if the drug is sneaky? What if it actually does manage to pick the lock, bypass the porins, and get inside the cell?
That brings us to the second mechanism, which is arguably one of the most aggressive strategies they employ: efflux pumps.
Ah, I was reading about these. I always picture these as incredibly aggressive bouncers at a nightclub. Yeah. The antibiotic manages to sneak past the velvet rope and get inside the club, but before it can even order a drink, or in this case, disrupt the cell's machinery, this massive bouncer literally grabs it by the collar and throws it right back out onto the street.
The bouncer analogy is actually spot on, primarily because these efflux pumps actively consume the cell's energy to forcibly expel toxic substances.
They actually use energy for this.
Yes, they are working hard to physically throw the drugs out. And these aren't just simple one-trick ponies. They are highly sophisticated transport proteins embedded deep in the bacterial membrane.
Okay.
The scientific literature categorizes them into several main families based on their physical structure and exactly how they source their energy. You'll see acronyms like the RND, MFS, SMR, ABC, and MADI families.
Whoa, okay, that's a lot of alphabet soup. How did these things actually get the power to physically push a chemical out of the cell?
Let's take the RND family as a prime example. These pumps utilize something called the proton motive force of the cell to power the mechanism.
Hold on, proton motive force. You just lost me. Are we talking about microscopic batteries here?
In a way, yes. The bacteria naturally maintain an imbalance of protons, positively charged particles across their membrane.
Okay, I'm with you.
There are more protons on the outside than the inside, which creates an electrochemical gradient. It's essentially stored energy, very much like a battery.
Right.
The R&D efflux pump allows protons to flow back inside the cell, and it uses the energy released by that flow to power the physical expulsion of the antibiotic molecule in the opposite direction.
That is wild. They are literally using an electrical charge across their skin to power a bouncer to throw our medicine away?
And the most notorious example of this is the Acri-B-Tulsi pump system found in Escherichia coli.
E. coli.
Yes. What makes this specific pump so devastating clinically is that it doesn't just recognize one drug. This single pump system can grab a massive variety of structurally completely unrelated antibiotics.
Like what?
Tetracyclines, chloramphenicol, fluorophenolones, it just pumps them all right back out.
Yeah.
It is a primary driver of multi-drug resistance because one single mechanism is capable of recognizing and throwing out almost every type of chemical weapon we deploy against it.
That is deeply unsettling. You could try 3 completely different classes of medicine and the same bouncer just keeps tossing them out.
Exactly.
Okay, so if changing the likes doesn't work and hiring bouncers doesn't work, what is the third mechanism?
The third is enzymatic degradation. Instead of just blocking the drug from entering or pumping it back out once it's inside, the bacteria go on the offensive.
They attack the drug itself.
Yes. They produce specific enzymes that actively seek out and chemically destroy the antibiotic molecule itself.
It's like sending out chemical scissors to chop up the key before it even hits the lock.
Precisely. And the most famous and frankly most problematic of these chemical scissors are the beta-lactamases.
Beta-lactamases.
Right. To understand how they work, you have to know that many of our most common antibiotics, penicillins, cephalosporins, carbapenems, all share a specific core structural feature called a beta-lactam ring.
Okay.
You can think of this ring as the active warhead of the antibiotic. It's the part that does the actual damage to the bacteria.
So the bacteria just target the warhead.
Yes. Bacteria like Klebsiella pneumoniae have evolved to produce specialized enzymes, such as KPC or the new Delhi metallo-beta-lactamase, also known as NDM.
NDM, right. I saw that in the reports.
These enzymes are designed to specifically target that beta-lactam ring. They chemically hydrolyze it, which means they literally break the ring open.
Snap it in half.
And once that beta-lactam ring is structurally broken, the entire antibiotic molecule is rendered completely inert. It's deactivated.
So it's just a dud floating around in the bloodstream while the bacteria just go about their business completely unharmed. That brings us to the fourth mechanism, which the sources call target modification. From my understanding, this is where the bacteria realize what specific internal part the antibiotic is aiming for, and they just change the target's shape so the drug can't recognize it anymore.
That's exactly right. Antibiotics work by binding to very specific proteins or enzymes within the bacteria to disrupt their function.
Like a lock and key.
Exactly. If the bacteria undergo a genetic point mutation that slightly alters the shape of that specific binding site, the antibiotic can no longer attach. A great example from our stack of research is MRSA, methicillin-resistant Staphylococcus aureus.
Everyone has heard of MRSA. It's the classic hospital superbug. How does it modify its target?
Normally, penicillins and methicillin work by binding to something called a penicillin-binding protein, which the bacteria use to build their cell walls.
Okay, so they need that protein to survive.
Yes. If the drug binds to it, the bacteria can't build its wall, and it dies. But MRSA has acquired a gene that allows it to produce an altered version of this protein, specifically called PBP2A.
PBP2A.
Right. This altered protein still functions perfectly well to build the cell wall, but its physical shape is just different enough that the antibiotic can no longer latch onto it.
It's like trying to put a square peg in a round hole, but the bacteria suddenly morphed a hole into a triangle. The drug just slides right off.
If we connect this to the bigger picture, you start to see why the World Health Organization and researchers worldwide call this a silent pandemic.
Yeah, silent but deadly.
Bacteria are not just passive, static targets waiting to be killed. They are highly reactive, deeply adaptive organisms. And they don't just act as isolated individuals floating around in your bloodstream.
Right, they team up.
One of the most formidable structural defensive strategies they employ is the formation of biofilms.
I saw a lot about biofilms in the literature. What exactly are they? Because when I think of bacteria, I just picture single microscopic pills floating around.
Most people do.
Yeah.
But you can think of a biofilm as a heavily fortified microbial city encased in a protective bunker.
A bunker.
When bacteria find a suitable surface, which could be the lining of your lungs, a heart valve, or a medical implant like a titanium knee, they attach to it and begin to collectively secrete an extracellular polymeric substance, or EPS.
Okay, stop right there. Extracellular polymeric substance. What does that actually mean in plain English? Is it like a shell?
It's more like a dense, slimy, three-dimensional shield made up of sugars, proteins, and DNA. It encases the entire colony of bacteria.
A microscopic slime bunker.
Yes.
So when you have an infection on a medical implant or in a chronic diabetic wound that just won't heal, it's not just free-floating bacteria. They are deeply entrenched in this slime.
Entrenched is the perfect word. And inside this EPS matrix, the bacteria become highly tolerant to our drugs for a few reasons.
Because the slime blocks the drug.
First, yes, the matrix itself acts as a physical barrier.
Yeah.
It literally slows down the diffusion of the antibiotic molecules, preventing them from reaching the bacteria buried deep inside.
Oh.
But more importantly, the environment inside the biofilm changes the bacteria's behavior. The ones deeper inside the bunker are starved of oxygen and nutrients, so they enter a dormant, slow-growing state.
Why does them being dormant matter? Don't antibiotics just kill bacteria on contact?
Not exactly. Many of our most effective antibiotics, like the penicillins we discussed earlier, are only lethal to actively dividing cells.
Oh, because they target the cell wall building process.
Exactly. They work by disrupting the cell wall synthesis process when the bacteria are trying to multiply. Because these deep layer bacteria, often called persister cells, are dormant and not actively building new cell walls, the antibiotic has almost no effect on them.
Wow.
The drug washes over the biofilm, maybe kills the few active growing cells on the outermost edge, but the core infection simply survives.
And then what happens?
The moment you finish your prescription and the antibiotic is out of your system, those persister cells wake up and rebuild the colony.
That is incredibly frustrating. So they have slime bunkers, bouncers throwing drugs out, changing the locks on the doors, and chemical scissors.
It's a robust defense.
But here's where it gets really interesting. And frankly, it's the part of our research today that gave me the chills. They don't just keep these brilliant survival strategies to themselves.
No, they don't.
They actually share them with each other. The sources detail a process called horizontal gene transfer, or HGT. I like to think of this as bacteria essentially passing cheat codes to each other via Bluetooth. It's the ultimate microbial sharing economy.
It is arguably the greatest, most dynamic threat we face in the resistance crisis. Before we understood horizontal gene transfer, we assumed bacteria could only become resistant through vertical transmission.
Meaning from parent to child.
Meaning a bacterium develops a random, lucky genetic mutation, survives the antibiotic, and then passes that trait down to its offspring when it divides.
Which takes a lot of time.
Exactly, that takes time. But through horizontal gene transfer, bacteria don't have to wait for a random, lucky mutation. They can acquire resistance on demand directly from their neighbors.
How are they physically doing this?
Yeah.
Because they don't have USB ports.
They do this through three main, highly effective methods. The first is transformation.
Transformation, okay.
When a resistant bacterium dies, it can glyze or burst open, releasing its naked DNA into the surrounding environment. Other living bacteria can literally scavenge that free-floating DNA, absorb it, and incorporate those resistance genes into their own genome.
That is morbid. They are looting the dead for survival traits.
It is entirely pragmatic from a biological standpoint.
True.
The second method is transduction. This involves viruses called bacteriophages, which we will discuss heavily later, that naturally infect bacteria.
Right.
Sometimes when these viruses replicate inside a resistant bacterium, they accidentally package a piece of the bacteria's resistance DNA instead of their own viral DNA.
Oh, a mix-up.
Exactly. When that virus goes on to infect a new bacterium, it inadvertently injects the resistance gene into the new host.
So a virus basically acts as an accidental courier service for superbug DNA. And what's the third method? The sources seem to suggest this was the most common one.
The third is conjugation, and it is the most direct and alarming method.
Why alarming?
In conjugation, two completely different bacteria physically approach each other. One of them extends a microscopic, tube-like appendage called a pilus to physically connect to the other.
Like a tunnel.
It's essentially a biological bridge. The donor bacterium then makes a copy of a plasmid, which is a small, circular, highly mobile piece of DNA that often contains multiple resistance genes and sends that copy straight through the pilus into the recipient bacterium.
Wait, so a completely harmless, everyday bacteria hanging out in your gut could literally walk up to a deadly invading pathogen, build a bridge, and hand it the exact genetic blueprint for surviving our strongest drugs?
Yes.
That's terrifying.
And what's worse, they don't just do this with their own species. They can transfer these plasmids across entirely different, unrelated species of bacteria. It is a completely open-source global network of resistance.
An open-source network of resistance. And this brings us to a crucial, deeply personal consequence of our antibiotic use, the collateral damage to the human microbiome.
Yes.
Because if we zoom out from the microscopic level, we realize that when you take a pill, it doesn't just go to your infected ear or your sinus, it goes everywhere. And we often prescribe what are called broad-spectrum antibiotics, right?
Yes. In clinical practice, if a patient presents with a severe infection, doctors don't always have the luxury of waiting 48 to 72 hours for a lab culture to tell them exactly which specific bug is causing the illness.
Right. The patient could die in that time.
Exactly. So they deploy a broad-spectrum antibiotic, which is chemically designed to indiscriminately kill a massive variety of bacterial species.
It's a shotgun approach.
It's a pragmatic necessity. But taking a broad-spectrum antibiotic is functionally akin to dropping a bomb on a dense forest, just to eliminate a single invasive weed. You destroy the weed, but you also completely annihilate the entire surrounding ecosystem.
And we need that ecosystem. Imagine you are listening to this right now. You have trillions of commensal bacteria, the good guys living inside your gut.
Trillions.
They help you digest your food, they produce essential vitamins, and they are essentially your body's microscopic real estate agents. They occupy the physical space and consume the available nutrients, so the bad guys, the pathogens, literally have nowhere to set up camp.
Exactly. The scientific term for that is competitive exclusion. Your commensal bacteria are your first line of defense.
Right.
When you take a broad-spectrum antibiotic, you wipe out that protective, flourishing forest of good bacteria. You leave the land entirely barren. And the clinical data on the consequences of this is absolutely staggering.
I know exactly what study you're going to mention.
We found a study in our research stack that perfectly illustrates this, looking at the infectious dose, or ID50, of Salmonella in mice.
Okay, so let's break this down for the listener because this study blew my mind. What does ID50 actually mean?
ID50 stands for the infectious dose required to successfully establish an infection in 50% of the test subjects.
Okay.
Normally, an animal, just like a human, has a robust, highly populated gut microbiome. The researchers found that under these normal, healthy conditions, you have to orally expose a mouse to an astonishing 10 million Salmonella cells just to force an infection to take hold.
10 million cells.
10 million.
That's a massive microbial army required just to break through the host's natural defenses and get past those real estate agents. It proves how good our bodies naturally are at fighting off everyday exposure.
It does.
But what happens after the antibiotic is introduced?
The researchers administered just a single standard dose of the antibiotic streptomycin to the mice. That single dose decimated their protective commensal microbiome.
The forest was burned down.
The forest was burned down. When the researchers retested the infectious dose of Salmonella on these antibiotic-treated mice, the number required to cause infection plummeted from 10 million cells to fewer than 10 cells.
Wait, less than 10?
Fewer than 10.
You're telling me that knocking out the good bacteria took the required invading force from 10 million down to single digits?
Yes.
So what does this all mean in a real-world scenario for someone listening right now? Let's say you have a totally healthy gut. You could practically eat a piece of slightly undercooked, contaminated chicken at a barbecue, and your body might just shrug it off entirely because your good bacteria outcompete the salmonella.
Very likely, yes.
But if you happen to be on a heavy course of antibiotics for a random, unrelated sinus infection, simply touching a poorly washed cutting board could land you in the emergency room.
That is precisely what the data implies, and it is a reality physicians see all the time. By chemically carpet bombing our internal ecosystem to treat one problem, we inadvertently make ourselves infinitely more vulnerable to secondary opportunistic pathogens.
We are actively clearing the land for the invaders.
We are.
That is a phenomenal, albeit terrifying, transition into the second major part of our discussion today, active debate, or where the science disagrees.
This is a complex area.
Because this entire idea that the cure might actually be causing massive secondary problems leads us to a paradox that is heavily debated in the medical literature. And it asks a deeply unsettling question. What if taking the antibiotic actually makes the bacteria more deadly to the patient?
This raises an important question regarding a phenomenon known as the bacterial SOS response.
The SOS response.
When bacteria are exposed to severe lethal stress, such as the sudden introduction of a toxic antibiotic, they don't just passively accept their fate and die quietly. They panic.
They have a panic mode.
From an evolutionary perspective, they activate a desperate emergency genetic network called the SOS response. This system halts normal cell division and rapidly upregulates genes designed to repair massive DNA damage, hoping to survive the assault.
Okay, so they go into full lockdown emergency repair mode. That makes sense. But why does that make them more deadly to us?
Because in certain highly dangerous pathogens, this genetic panic response is directly hardwired to their virulence factors.
Virulence factors meaning?
Meaning their biological weapons. A terrifying real-world example detailed in the research involves a very specific strain of bacteria, Escherichia coli O157H7.
O157H7.
This is a strain famous for causing massive, widespread foodborne illness outbreaks, often linked to contaminated leafy greens or undercooked beef.
Right. This is the one you hear about on the news that forces massive national food recalls, and it produces something called the Shiga toxin, correct?
Yes. The Shiga toxin is one of those potent bacterial poisons known to science.
Really?
When it enters the human bloodstream, It aggressively binds to receptors on human cells, particularly in the kidneys and the brain, and completely shuts down cellular protein synthesis.
Which does what?
This leads to massive cell death, severe kidney damage, a condition called hemolytic uremic syndrome, and very often death.
Okay, so this bug is a nightmare. What happened in the study when they tried to kill it with antibiotics?
Researchers tested what happens when you expose this specific E. coli strain to a common, highly effective fluoroquinolone antibiotic called ciprofloxacin.
A standard drug.
Yes, the antibiotic worked exactly as designed. It attacked the bacteria and successfully triggered massive DNA damage.
Okay, good.
This immediately induced the bacterial SOS response. But in doing so, the panic response didn't just try to repair the DNA. It inadvertently triggered a massive overdrive in the transcription of the genes that produce the Shiga toxin.
Oh, no.
The result was a 17-fold increase in the production of the lethal Shiga toxin compared to bacteria that were left completely alone.
A 17-fold increase. So the drug is basically triggering the bacteria's self-destruct sequence, but the bacteria's response is to pull the pin on a massive biochemical grenade just before it dies.
That is a very visceral but highly accurate way to describe the mechanics of it. In the animal models detailed in the study, the mice that were given the antibiotic experienced a massive sudden spike in circulating Shiga toxin levels in their system.
And what happened to the mice?
The horrifying outcome was that the mice with the highest toxin levels died after receiving the antibiotic. The drug successfully lowered the total bacterial count. It killed the bug. But the resulting panic-induced toxin release was fatal to the host.
Hold on. If giving the antibiotic makes the E. coli panic and release a fatal amount of toxin, are you saying doctors are supposed to just sit there and do nothing?
It's incredibly difficult.
How on earth do you treat a patient if the very cure is what kills them?
That is the exact core of the clinical debate. It creates a massive, agonizing dilemma for a physician treating a patient with this specific infection. If you give the antibiotic to stop the bacteria from multiplying and spreading, you know you risk triggering a fatal systemic toxin dump.
Right.
If you withhold the antibiotic, the bacteria continue to multiply unchecked. In many cases of Shiga toxin-producing E. coli, the standard care is actually supportive therapy.
Meaning what?
Meaning 4V fluids, managing blood pressure, and supporting the kidneys. While explicitly avoiding antibiotics, allowing the immune system to clear the infection naturally without triggering the SOS response.
That is an impossible choice to have to make as a doctor. And the crazy thing is, it's not just the Shiga toxin that poses this threat.
No, it isn't.
Our sources talk about something called endotoxins and a phenomenon known as the Jarisch-Herxheimer reaction. Let's unpack this. Because if the bacteria don't actively secrete a toxin when they panic, they might just naturally be full of toxins that violently spill out when we blow them up with drugs, right?
Exactly. We have to look at the physical architecture of gram-negative bacteria again. They have a complex molecule embedded right into their outer membrane called lipopolysaccharide, or LPS.
LPO.
The lipid, or fat, portion of this molecule is known as lipid A, and it is fundamentally an endotoxin. It is physically part of the bacteria's structural skin.
Oh, okay.
Now, as long as the bacterium is alive and physically intact, that toxin is relatively contained and managed by the immune system in small doses.
But when we introduce a heavy-duty antibiotic...
When an antibiotic, particularly a beta-lactam like penicillin that targets the cell wall, successfully attacks and lyses or literally pops the bacterial cell, the bacterium explodes.
Right.
Its physical structure fragments, releasing massive, overwhelming amounts of these embedded endotoxins directly into the patient's circulating bloodstream all at once.
It's like shooting a toxic water balloon. You pop the balloon, and the poison just spills out everywhere, flooding the system.
And the human immune system reacts violently to a sudden flood of free-floating endotoxins. It perceives an overwhelming systemic invasion and triggers a massive full-body inflammatory response.
What does that look like in a patient?
The patient can suddenly experience severe, spiking fevers, uncontrollable chills, a dangerous rapid drop in blood pressure, and in severe cases, fatal endotoxic shock. This intense physiological crash following antibiotic administration is called the Jarisch-Herxheimer reaction.
It perfectly demonstrates the profound paradox of modern antibiotic therapy. The very act of successfully destroying the pathogen can inadvertently kill the host. It's a terrifying tightrope to walk.
It really is.
And this debate around how aggressively we should attack these infections brings us to a huge controversy in the clinical world over broad-spectrum versus narrow-spectrum drugs, and specifically the use of combination therapies.
Right. Combination therapies.
If one single drug isn't working because of resistance, doctors will often mix multiple drugs together into a cocktail, right?
Yes, it is a very common clinical strategy designed to completely overwhelm the pathogen's defenses. If the bacteria have evolved a shield, you send in one drug to break the shield and another to deliver the lethal blow.
Can you give an example?
A common, heavily utilized example is combining a standard beta-lactam antibiotic, like ceftolozane, with a specific beta-lactamase inhibitor, like tazobactam.
Okay, so how do those two drugs work as a team?
The tazobactam essentially acts as a sacrificial decoy. It intentionally binds to and permanently disables those bacteriochemical scissors, the enzymes we discussed earlier that the bacteria use to destroy the antibiotic.
Oh, brilliant. So it distracts them.
By neutralizing the scissors, the tazobactam clears a safe path, allowing the primary antibiotic, the ceftolozane, to slip past the defenses unharmed and successfully destroy the bacterial cell wall. Clinically speaking, it is highly effective at clearing stubborn resistant infections.
But there is a massive catch.
There's a significant, heavily debated catch. While combination therapies successfully overwhelm the target bacteria, they also apply a devastating compounded amount of selective pressure to the patient's entirely healthy microbiome.
Forest fire again.
It's the forest fire analogy we used earlier, but severely amplified. Furthermore, there is intense ongoing debate in the nephrology community about the physiological toll these chemical cocktails take on the human body's filtration systems.
Right. The data we found on this in the stack is alarming. One major study highlighted that using combination therapies like piperacillin tazobactam increased the patient's risk of developing acute kidney disease by over 150% compared to just using a single monotherapy drug.
Over 150%.
So what does this all mean for the patient lying in the hospital bed? You successfully cure their superbug pneumonia, but you might leave them requiring dialysis for the rest of their life.
It is a delicate, high-stakes balancing act of toxicity versus efficacy. And this debate extends far beyond just the administration of the drugs. It bleeds heavily into how we help patients recover from these intensive microbiome-destroying treatments.
Right, the aftermath.
Once that internal forest is burned down by the combination therapy, how do we ethically and effectively rebuild it? This brings us to the massive billion-dollar controversy surrounding probiotics and prebiotics.
Ah, the recolonization debate. Walk into any pharmacy or grocery store in the world, and you'll see an entire wall of beautifully packaged probiotics promising to restore your gut health after a round of antibiotics.
You certainly will.
You hear influencers touting them constantly. Do they actually work, or is it just highly expensive marketed yogurt?
The clinical data on this is incredibly mixed, which is exactly why it remains such an active, heated debate among gastroenterologists and microbiologists. The underlying ecological theory is logically sound. If the forest is burned down, you need to replant seeds.
Makes sense on paper.
You introduce massive quantities of beneficial bacteria to rapidly outcompete any lingering pathogens and restore the natural ecosystem. And there are specific, highly documented clinical successes. For instance, the literature extensively details the clinical use of a specific strain, Lactobacillus rhamnosus GG.
How is that specific strain used?
It was used in trials to successfully decolonize hospitalized patients who were harboring vancomycin-resistant enterococci, or VRE, in their guts.
What is VRE, and why is it so dangerous?
Enterococci are bacteria that normally live harmlessly in our intestines. But in hospital settings, especially after heavy antibiotic use, they can become highly resistant to vancomycin, which is one of our most powerful last line of defense antibiotics.
The glass box drugs.
Exactly. If VRE escapes the gut and gets into the bloodstream or a surgical wound, it causes massive untreatable infections. In these specific trials, doctors gave the patients heavy targeted doses of the Lactobacillus rhamnosus GG probiotic.
What did the probiotic do?
The probiotic successfully moved into the gut, aggressively consumed the local nutrients, and physically crowded out the dangerous VRE, eventually clearing it from the patient's systems.
So in that specific, highly controlled case, it worked brilliantly as a targeted ecological therapy. The good guys forced the bad guys out.
Yes, but the variability in clinical success across the broader general population taking off the shelf probiotics is massive. The science is incredibly messy.
Messy how?
Some large-scale studies show absolutely no statistical benefit compared to a placebo. Other, more recent studies suggest something even more concerning. that taking a concentrated commercial probiotic might actually delay the natural recovery of your microbiome.
Wait, taking the probiotic makes it take longer to heal? How is that possible?
Because a healthy human gut is an incredibly diverse rainforest containing thousands of different interacting species. When you take a commercial probiotic pill, you are often flooding the system with a massive, overwhelming monoculture of just one or two specific bacterial strains.
Ah,
You're planting millions of identical pine trees. This massive artificial influx can actually suppress the natural, slow reestablishment of the diverse flora that we're trying to grow back natively. It is far from a settled science, and the marketing vastly outpaces the clinical consensus.
Okay, so let's summarize where we are. We've established that our current foundation of modern medicine is severely cracking.
It is.
The golden era of penicillin is long over. The bacteria are evolving and sharing resistance genes faster than we can invent new chemicals. The broad-spectrum drugs are destroying our protective microbiomes. And sometimes the physical act of killing the bug triggers a panic response that kills the patient. It paints a pretty bleak, terrifying picture.
It does. But we cannot stop the discussion there.
Exactly. Which brings us to part 3 of our exploration today, frontiers. What might change absolutely everything? Because human ingenuity hasn't stopped, and brilliant scientists around the world are looking far, far beyond traditional chemical antibiotics. Let's talk about phage therapy. I call this the enemy of my enemy is my friend approach.
Bacteriophages are arguably one of the most fascinating, alien-looking entities in all of biology. To visualize them, imagine a microscopic lunar lander, complete with a geometric head and spindly landing legs.
I've seen pictures of these. They look like tiny robots.
They do. They are viruses, but they are completely, fundamentally harmless to human cells. They have absolutely zero interest in us. They exclusively hunt, infect, and kill bacteria. They are the apex natural predators of the microbial world, and they have been locked in a relentless evolutionary arms race with bacteria for billions of years.
So instead of throwing a dumb chemical bomb into the body and hoping for the best, we're intentionally sending in a biological assassin.
Exactly. And the clinical advantages of this approach are profound. Because phages have evolved alongside bacteria, they are highly, incredibly specific. They don't just kill any bacteria. A specific phage often targets only a single specific strain of a single species of bacteria.
They act like a microscopic sniper rifle rather than a shotgun.
Yes.
So how does that benefit the patient's microbiome?
If a patient has a severe multi-drug-resistant Staphylococcus aureus infection, doctors can administer a phage specifically evolved to hunt that exact strain of staph.
Okay.
The phage will enter the body, ignore the human cells, ignore the millions of beneficial commensal bacteria in the gut, and strictly hunt down the staph bacteria. There is absolutely zero collateral damage to the host's internal ecosystem.
That is incredible. But what about the biofilms we talked about earlier? Those slime bunkers that antibiotics can't penetrate? Can phages get through those?
That is one of their most powerful advantages. Remember, phages have been fighting bacteria for billions of years. They have encountered biofilms before.
Right, they know the tricks.
Many phages have naturally evolved specialized, highly potent enzymes specifically designed to break down and dissolve the EPS matrix of bacterial biofilms. They can literally melt the roofs of the bunker, swim directly inside, and begin slaughtering the dormant persister cells that chemical antibiotics simply cannot reach.
That is mind-blowing. Nature already built the perfect weapon. But here's where it gets really interesting, and where the stack of biotech journals we read crosses from brilliant biology into what feels like pure science fiction.
We are definitely entering a new era.
Scientists aren't just taking wild phages from a puddle or a river anymore. They are upgrading them in the lab. The sources detail CRISPR-Cas engineered phages. When I read this, my jaw hit the floor.
It truly is a monumental paradigm shift in medical science. To understand the magnitude of this, we must first remind ourselves of what CRISPR actually is. It has become a massive buzzword, but CRISPR was not invented by human beings in a laboratory.
Wait, it wasn't?
It is, ironically, the ancient natural bacterial immune system.
Bacteria have an immune system.
Yes. For billions of years, bacteria have used the CRISPR-Cas system to memorize the specific DNA sequences of invading bacteriophages.
Okay.
If that specific phage ever tries to infect them again, the bacteria use the Cas proteins to recognize the viral DNA and chop it up, neutralizing the threat. What human scientists have brilliantly done is hijack this natural bacterial defense system and engineer it into a highly targeted offensive weapon.
So we are taking the bacteria's own biological shield, pulling it out of them, forging it into a sword, and handing it back to the viruses to use against the bacteria.
That is beautifully put.
How does this actually work in a real clinical setting?
Let's look at the cutting-edge clinical trials detailed extensively in our sources. There is a biotech company called Locus Biosciences that is currently in advanced phase 3 clinical trials for a drug designated as LBPEC01.
LBPEC01.
Got it. This is an in vivo CRISPR phage therapy specifically targeting multi-drug-resistant E. coli that causes severe recurring urinary tract infections.
Okay, break down the mechanics of this for me. What exactly did they build?
They've taken a natural bacteriophage that already naturally infects E. coli, and they have genetically engineered a highly specialized CRISPR-Cas3 construct directly into its payload.
Okay, let's unpack this payload. Everyone has heard of CRISPR-Cas9 in the news for gene editing. What does Cas3 do differently?
It's a vital distinction. While the famous CRISPR-Cas9 acts like a pair of molecular scissors, making a single, highly precise, repairable cut in a DNA strand to edit it, Cas3 is an entirely different beast.
How so?
Cas3 acts like a molecular wood chipper. Once it binds to the target DNA, it progressively travels down the strand, aggressively chewing it up and absolutely shredding the bacterial genome beyond any hope of repair.
That is brutal. So the engineered phage lands on the outside of the multi-drug-resistant E. coli, injects this CRISPR-Cas3 machinery inside, and the machinery specifically identifies the bacteria's core DNA and completely destroys it from the inside out.
Yes. The bacteria's own internal blueprints are systematically shredded, leading to rapid, guaranteed cell death.
It's like sending a highly sophisticated, precision-guided drone that delivers a corrupted, lethal software update exclusively to the bad guys. It causes them to completely self-destruct while leaving the innocent bystanders, our good bacteria, entirely, blissfully alone. That is breathtaking science.
It really is.
And Locus isn't the only company pushing this envelope, right?
Far from it. The literature also highlights SNIPR Biome, which recently completed a phase 1 clinical trial for a therapy called SNIPR-001.
What does that one do?
This therapy takes a slightly different, but equally brilliant approach. It uses 4 entirely different engineered bacteriophages to deliver a CRISPR-Cas9 payload to selectively eliminate dangerous strains of E. coli directly from the human gut, specifically aiming to prevent severe blood infections in leukemia patients.
Why use 4 different phages at once? Isn't one enough?
It's a strategic move to preempt the bacteria's ability to adapt. If you send in just one type of phage, the bacteria might quickly mutate its outer receptors to block that specific virus from landing.
Ah, right, changing the locks again.
Exactly. By using 4 completely different phages simultaneously, targeting 4 different landing sites, they're preventing the bacteria from easily developing a resistance mechanism to the delivery vehicle itself. It's a highly sophisticated, multi-pronged biological attack.
So we are literally moving from carpet-bombing chemical warfare to programmatic, hyper-targeted genetic warfare. But what if we don't want to kill the bacteria at all? The sources mention an entire field called antivirulence therapeutics. This concept completely blew my mind. The entire idea is disarming instead of killing.
This is a radical, deeply philosophical, and highly logical new approach to infectious disease. We have to look at the root cause of the antibiotic resistance crisis. The root cause is the extreme life-or-death selective pressure we apply.
Right, the evolutionary boot camp.
If you spray a toxic chemical that kills 99.9% of a population, the 0.1% that survives because of a random mutation is absolutely guaranteed to take over. It's an existential battle for survival.
Yeah.
But what if we don't threaten their actual life? What if we just take away their weapons?
Because if they aren't actively dying, they aren't under massive evolutionary pressure to rapidly mutate and share resistance genes, they don't panic.
Exactly. You remove the selective pressure. One of the primary fascinating methods being explored in this field is called quorum quenching.
Quorum quenching.
To understand it, we need to know that bacteria do not just act as isolated, deaf, and blind individuals. They constantly communicate with each other using secreted chemical signals in a highly coordinated process called quorum sensing.
They talk to each other.
Chemically, yes. They secrete signaling molecules into their environment, and they also have receptors to detect the concentration of those molecules. It allows them to gauge their own population density.
Like taking a roll call.
Yes. Once their population reaches a certain critical density, a quorum, they collectively, simultaneously alter their gene expression to launch an attack. They decide, as a unified group, to start secreting deadly toxins, or to begin building that impenetrable biofilm bunker we discussed.
That is terrifyingly coordinated. So what does quorum quenching do to stop that?
Quorum-quenching therapeutics are specifically designed to intercept, degrade, or scramble those chemical signals. They introduce enzymes that chop up the signaling molecules, or they introduce antagonist molecules that block the bacteria's receptors.
I love this. It's exactly like jamming the radio communications of an invading army. The soldiers are all still physically standing there on the battlefield, fully armed, but their radios are just playing static.
That's a great analogy.
They don't know how many of them are out there, they don't know when the general is ordering them to attack, and they absolutely can't coordinate to build their fortresses.
And the beauty of this is what happens next. Because they are confused, uncoordinated, and exposed, but not actively being killed by a toxic drug, they don't trigger that SOS panic response.
So no toxic chemical dump.
Right. The human host's natural immune system, which is usually overwhelmed by toxins or blocked by biofilms, can simply sweep in like a cleanup crew and clear the infection away normally. You entirely neutralize the threat without ever triggering the desperate evolutionary panic that inevitably leads to superbugs.
Think about what a monumental shift that is. For almost 100 years, humanity's only medical strategy against bacteria has been search and destroy. Moving to a philosophy of search and disarm is a profound leap in how we interact with the natural world.
It truly is.
We are also seeing a major shift toward using monoclonal antibodies, or mAbs, to achieve this, right? We've heard a lot about mAbs in the context of cancer or autoimmune diseases, but how are they being deployed against bacteria?
Monoclonal antibodies act as ultra-precision biologics. Instead of targeting the physical bacterial cell itself to kill it, they can be perfectly synthesized in a lab to target and bind to the specific lethal toxins the bacteria produce. A prime, highly successful example heavily featured in our sources is bezlotoxumab.
Let's look at bezlotoxumab. How does it work?
This is an FDA-approved monoclonal antibody specifically designed to bind to and neutralize toxin B, which is the primary destructive toxin produced during severe Clostridioides difficile, or C. diff, infections.
Ah, C. diff. That is notorious for destroying the colon lining, usually after someone has taken a ton of broad-spectrum antibiotics and wiped out their gut flora.
Exactly. So if a patient has a severe C. diff infection, the bacteria are aggressively pumping out this toxin B, which causes massive inflammation and tissue damage in the colon.
And the antibody stops that.
When you administer bezlotoxumab, the massive influx of antibodies directly binds to the toxin molecules floating in the gut and completely neutralizes them before they can attach to the human cells and do any damage.
So you aren't necessarily killing the C. diff bacteria with the antibody. You are just rendering its primary weapon completely harmless.
Precisely. It provides rapid, targeted relief, prevents the severe, life-threatening symptoms of the disease, and crucially, it does this without acting as a broad-spectrum killer that would further devastate the already fragile gut microbiome. It is a much more elegant surgical approach to infectious disease management that respects the underlying ecology of the human body.
These frontiers, the phages, the CRISPR wood chippers, the radio jamming, the antibodies are incredibly exciting, and they give me a lot of hope.
They should. The science is amazing.
But we have to face reality. Many of these are still in clinical trials or early stages of adoption. Which brings us to our final and perhaps most sobering segment today, implications. We need to look at the ethics, the economics, and society at large.
This is where the rubber meets the road.
Because while the science is advancing rapidly in the lab, the real-world toll happening right now in hospitals around the globe is absolutely devastating. Let's look at the sheer numbers.
The global toll of antimicrobial resistance is no longer some distant theoretical future projection. It is a brutal, present reality. The most comprehensive, rigorously vetted analyses we have indicate that if we do not drastically alter our current trajectory, up to 10 million people could die annually from resistant infections by the year 2050.
Let that number sink in. 10 million people every single year.
To put that staggering number in perspective, that would surpass the annual deaths currently caused globally by all forms of cancer combined.
It's horrifying.
Furthermore, the economic impact is almost unfathomable. The projections suggest a $100 trillion loss in global GDP by 2050.
100 trillion.
This is due to massive losses in workforce productivity, massively prolonged hospital stays requiring intensive care, and completely exorbitant skyrocketing healthcare costs as cheap, generic pills are replaced by incredibly expensive last resort interventions.
$100 trillion. It is a scale of loss both in human life and economic stability that is almost impossible to comprehend. And to try and manage and prioritize this escalating crisis, the World Health Organization just updated their highly influential Bacterial Priority Pathogens list for 2024.
Yes, the BPPL.
What does this massive new list actually tell us about who is currently winning the war right now?
The 2024 WHO BPPL is a deeply critical strategic document for global health. It rigorously evaluated 24 specific, highly dangerous pathogens across 15 different bacterial families. They ranked them using extremely strict criteria, including global mortality rates, treatability, and the current state of the pharmaceutical drug development pipeline. The list is divided into tiers to guide global research funding.
And what is sitting at the very top? What is keeping public health officials awake at night?
The critical priority group, the absolute top tier, remains entirely dominated by highly adaptable gram-negative bacteria. This includes carbapenem-resistant Enterobacterales, often clinically referred to as CRE, and carbapenem-resistant Acinetobacter baumannii, or CI Brau.
I've heard doctors call those the nightmare bacteria.
They absolutely are. These are the pathogens that routinely tear through hospital intensive care units. They are essentially pan-resistant. Meaning they are resistant to almost everything we throw at them, including our absolute last resort, highly toxic drugs. The mortality rates when these infect the bloodstream are catastrophic. The critical list also prominently features rifampicin-resistant tuberculosis, which poses a massive, unique global health security threat simply due to its rapid airborne transmission in densely populated areas.
So the critical list is the imminent hospital and airborne threats.
Yeah.
What about the next tier down on the WHO report?
The high priority group highlights pathogens that, while maybe slightly more treatable than the critical tier, cause absolutely massive global disease burdens, often spreading rapidly in everyday community settings.
Like food poisoning.
Yes, exactly. This includes fluoroquinolone-resistant Salmonella and Shigella, which severely complicate the treatment of life-threatening food and waterborne illnesses, especially in developing nations. It also includes our old, highly adaptable nemesis, MRSA methicillin-resistant Staphylococcus aureus, and increasingly drug-resistant Neisseria gonorrhea.
Gonorrhea.
Yes, which is truly terrifying because it threatens to make a very common, easily spread sexually transmitted infection entirely, permanently untreatable.
So what does this all mean for the way we actually handle this crisis as a global society? We clearly can't just look at human patients in a hospital bed in isolation. Our sources lean incredibly heavily into a paradigm called the One Health perspective.
One Health perspective is essential.
Okay, let's unpack this concept thoroughly, because it essentially argues that you cannot possibly solve human antibiotic resistance without looking at the cow in the field and the river flowing behind the hospital.
The One Health perspective is the fundamental, vital recognition that human health, animal health, and environmental health are not separate silos. They are deeply, inextricably linked in a continuous cycle. We absolutely cannot solve the AMR crisis by only focusing on human clinical medicine. We have to look at agriculture.
Agriculture uses a lot of antibiotics, right?
A staggering amount. The almost incomprehensible volume of antibiotics used in global agriculture is a primary driver of resistance.
And they aren't even using these drugs just to treat sick animals, are they?
That is the most alarming part. A massive percentage of agricultural antibiotics are administered daily at sublethal doses directly into the feed and water of entirely healthy livestock, pigs, chickens, cattle, simply to act as artificial growth promoters to get them to slaughter weight slightly faster.
Wow.
This massive chronic low-dose exposure creates the perfect, vast reservoirs for resistance genes to rapidly evolve and multiply in the guts of millions of animals.
And those genes don't just stay inside the cow?
Exactly. These highly concentrated resistance genes, along with active, unmetabolized antibiotics, are then excreted by the ton into the environment, deeply contaminating agricultural soil, groundwater, and major waterways.
Which brings me to a statistic about human infrastructure that really stuck with me from the reading. Yeah. You mentioned hospital wastewater earlier. There was a study noting a 100% incidence rate of the antibiotic effluxacin in the effluent of some wastewater treatment plants.
Yes, 100%.
100%. That means we are literally marinating the naturally occurring bacteria in our municipal sewage systems in constant sublethal doses of our best drugs. We are operating taxpayer-funded global training facilities for superbugs right under our own streets.
It is a profound infrastructural failure. And this intense environmental contamination inevitably spills over into global wildlife. Studies detailed in our sources have shown wild birds, migratory waterfowl, and mammals carrying clinically relevant, highly resistant human pathogens in their guts.
They carry it with them.
They act as rapid, highly mobile biological vectors, spreading these dangerous resistance genes across vast international geographical distances in a matter of days. If we do not aggressively address the environmental and agricultural pillars of the One Health triad, any brilliant billion-dollar advancements we make in human clinical medicine will inevitably be outflanked by nature.
That is a terrifying reality to face, but arguably the most frustrating head-against-the-wall part of this entire exploration today is what the sources call the economic market failure of antibiotics.
The economics are deeply flawed.
We desperately need new drugs. We have brilliant scientists who know how to make them. So why are the massive multi-billion-dollar pharmaceutical companies completely abandoning the field? The data in this stack is stark. 78% of major pharmaceutical companies entirely abandoned antibiotic research and development between 1990 and 2019. Why?
It is perhaps the most tragic, deadly irony of modern capitalist economics. Developing a completely new, novel class antibiotic one that works differently than the penicillins or the fluoroquinolones is incredibly difficult, scientifically risky, and astronomically expensive.
How expensive?
It typically takes 10 to 15 years of grueling lab work and clinical trials, and upward of $1 billion to $1.5 billion in research and development funding just to bring a single new antibiotic safely to market.
Okay, $1 billion is a massive investment. But let's play this out. Let's say a biotech company successfully does it. They spend the $1 billion, they run the gauntlet of the FDA, and they create a brilliant new drug that successfully kills the nightmare, critical priority superbugs on the WHO list. What happens next? They should make billions, right?
In a normal pharmaceutical market, yes. Sure. But antibiotics are unique. When that brilliant new drug hits the hospital formulary, the infectious disease stewardship committees get hold of it. And from a public health perspective, rightfully so. They take this shiny new billion-dollar drug and they essentially lock it in a glass box on the wall that says, break only in case of absolute catastrophic emergency.
Keep it away from everyone.
They mandate to all the doctors, do not prescribe this new drug under any circumstances unless every other older generic drug has completely failed, because we absolutely do not want the bacteria developing resistance to our only new weapon.
So the drug is a massive, triumphant success scientifically, but a complete and utter disaster commercially.
Exactly. Because the drug is so precious that it is held in extreme reserve, the actual sales volume, the number of pills being bought by hospitals, is practically zero. The company physically cannot sell enough pills to ever recoup its billion-dollar investment, let alone make any profit for the shareholders who funded the research.
Right, because traditional pharma relies on selling tons of pills.
The traditional pharmaceutical business model is entirely volume-driven. You make billions of dollars by selling millions of pills every single day for decades to treat chronic, ongoing conditions like high cholesterol, depression, or diabetes.
But antibiotics are the exact opposite of a chronic treatment. They're meant to be taken for 7 days, completely cure the patient, and then you stop taking them.
Precisely. When you combine the inherently short duration of the treatment course with the incredibly strict stewardship protocols severely limiting their use, the economic return on investment completely collapses.
Has this actually happened? Companies going bankrupt after inventing a drug?
We have seen incredibly sad, highly publicized cases where small, wildly innovative biotech companies successfully achieved the impossible they went from. They brought a powerful new antibiotic to market only to declare absolute bankruptcy just months later because literally no one was allowed to buy the drug.
So the free market is fundamentally completely broken when it comes to antibiotics. We can't rely on normal capitalism to save us here. We have to fundamentally change the way we pay for these drugs. What are the actual proposed solutions? The sources spent a lot of time talking about something called pull incentives.
Pull incentives are a radical shift in healthcare economics, specifically designed to completely decouple the profitability of a drug from the actual volume of sales.
Okay, so you don't pay per pill.
Instead of paying a company per pill sold, the idea is to legally guarantee a massive upfront return on investment for any company that successfully develops a highly needed, innovative antibiotic that targets the most critical superbugs. In the United States, there is currently proposed highly debated legislation called the PASTEUR Act, which would fundamentally create large subscription-style contracts between the government and the drug makers.
The sources refer to this as the Netflix model. I love this analogy, but explain it to me. How does a Netflix subscription work for a life-saving chemical?
Under the Netflix model, which is actually currently being piloted and tested in the United Kingdom, The government acts as a massive subscriber. They evaluate the new antibiotic, see its value to public health, and pay the pharmaceutical company a flat, massive, upfront subscription fee.
Like a yearly fee.
For example, 10 million or 20 million pounds a year for guaranteed, unlimited access to the antibiotic for their citizens. Crucially, the company gets that guaranteed, stable revenue to recoup their massive R&D costs, regardless of whether 1,000 pills are actually prescribed that year or absolutely zero.
So the company is happy because they don't immediately go bankrupt after inventing a miracle. And the hospital doctors are happy because they can keep the drug locked securely in the emergency glass box to prevent resistance without destroying the company that made it. The financial profit is totally 100% diverse from the volume of sales.
Precisely. It fundamentally incentivizes the creation of the antibiotic, not as a consumer good to be sold in bulk, but as a critical infrastructure insurance policy for society, much like a fire department.
But there has to be pushback on this, right? Are there critics who say governments shouldn't just be handing 10 million a year of taxpayer money to a giant pharmaceutical company for a drug that just sits on a shelf collecting dust?
Absolutely. It is highly debated. Critics argue that handing massive upfront payments to an industry already perceived as highly profitable is politically toxic. They worry about the government overpaying for drugs that might never actually be needed on a large scale, or that pharmaceutical companies might exploit the system by slightly modifying old drugs just to secure a lucrative subscription contract.
Right.
But the counter-argument from public health experts is stark. What is the alternative? If we don't pay up front to ensure these drugs exist, the pipeline will remain empty, and when the next massive, untreatable superbug outbreak hits, the economic and human costs will be immeasurably higher than any subscription fee. It is a necessary, albeit complex, evolution of healthcare economics if we want to ensure a steady pipeline of new therapies against the constantly evolving threat of AMR.
This has been an absolutely staggering journey through the microscopic world today. We've seen the ancient, prehistoric origins of resistance, the incredible, highly coordinated defensive fortresses bacteria build, the terrifying paradoxes where our current treatments might actually kill the patient, and the incredibly hopeful science fiction frontiers of CRISPR wood chippers and engineered phages. Before we wrap up today, looking at this massive stack of research, what is the final provocative thought you want to leave with the listener?
If we connect all of this to the bigger picture, the thought I want to leave you with is this. As we actively engineer viruses to seek out and edit bacterial DNA inside our own bodies using CRISPR, we are stepping into a profound, unprecedented new era of human history. We are finally moving away from the blunt force era where we chemically carpet bomb our biology and stepping into an era where we program it. But this raises an incredibly important and sobering question. Will the bacteria simply learn to hack our highly advanced CRISPR tools just as easily as they hacked penicillin 80 years ago? We must remember they are the ultimate survivors on this planet, and their genetic plasticity and ability to share information is completely unmatched. The arms race isn't over. It has just moved from the chemical level to the genetic level. And as a society, we must remain endlessly vigilant.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. We hope you enjoyed today's discussion. If you did, be sure to subscribe and support us on Patreon. And of course, you can write to us in comments if a specific topic needed to be covered, which help us all to grow. Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.